Hi. Today I'm going to tell you 100 sleepy facts about DNA, perfect if you want to relax a bit and learn something interesting about this amazing molecule of life. Enjoy listening. 1. DNA stands for deoxyribonucleic acid. DNA is the molecule that carries the genetic instructions for life. It is composed of long chains of nucleotides, which include a sugar, phosphate group, and a nitrogenous base. DNA determines traits and functions in all living organisms. Its structure allows it to store information efficiently. 2. DNA is shaped like a double helix. The famous double helix structure was discovered by James Watson and Francis Crick in 1953. It consists of two strands wound around each other, resembling a twisted ladder. The sugar-phosphate backbone forms the sides, while paired nitrogen bases form the rungs. This shape is crucial for replication and stability. 3. DNA contains 4 bases, A, T, C, and G, adenine, A, thymine, T, cytosine, C, and guanine, G, form the building blocks of DNA. A pairs with T, and C pairs with G, forming complementary base pairs. These pairs are held together by hydrogen bonds. The sequence of these bases encodes genetic information. 4. The order of bases determines genetic code. DNA sequences act like instructions for building proteins. Each set of 3 bases, called a codon, specifies a particular amino acid. Proteins are essential for cell structure, function, and regulation. Mutations in the sequence can alter protein function, sometimes causing diseases. 5. DNA is found in the nucleus of eukaryotic cells. In humans and other eukaryotes, most DNA is stored in the cell nucleus. A small amount also exists in mitochondria, called mitochondrial DNA. Nuclear DNA is responsible for most genetic traits. Its compartmentalization protects it from damage and organizes replication. 6. DNA is replicated before cell division. Replication ensures that each new cell receives an identical copy of DNA. Enzymes like DNA polymerase add nucleotides to create complementary strands. This process is semi-conservative, meaning each new DNA molecule contains one old and one new strand. Accurate replication is essential for growth and reproduction. 7. DNA mutations can affect traits. Mutations are changes in the DNA sequence. They can be caused by errors in replication, radiation, or chemicals. Some mutations are harmless, while others can alter proteins and lead to genetic disorders. Mutations also drive evolution by introducing genetic diversity. 8. DNA stores hereditary information. DNA is passed from parents to offspring. It carries genes, which determine traits like eye color, hair type, and susceptibility to diseases. This hereditary function allows species to maintain continuity across generations. DNA stability over time ensures reliable transmission of information. 9. DNA can be extracted from almost any cell. Scientists can isolate DNA from blood, hair, saliva, and even ancient bones. Extraction involves breaking cell membranes and separating DNA from proteins. This ability is essential for forensic analysis, genetic testing, and research. Even small amounts of DNA can be sufficient for study. 10. DNA makes RNA through transcription. DNA is used as a template to create RNA, which carries the instructions for protein synthesis. This process is called transcription. RNA then travels from the nucleus to the cytoplasm. This is the first step in the flow of genetic information, known as the central dogma. 11. DNA mutations can lead to cancer. Mutations in genes that control cell growth can result in uncontrolled cell division. Oncogenes and tumor suppressor genes are common targets. If DNA repair mechanisms fail, mutations accumulate, potentially causing tumors. Understanding DNA mutations is key in cancer research and treatment. 12. DNA is composed of nucleotides. Each nucleotide consists of a phosphate group, a sugar, deoxyribose, and a nitrogenous base. Nucleotides link together via phosphodiester bonds to form long chains. The sequence of nucleotides encodes genetic instructions. Nucleotide structure is fundamental to DNA stability and function. 13. DNA was first discovered in 1869. Swiss chemist Friedrich Miescher isolated a substance from white blood cells that he called nuclein. This substance was later identified as DNA. However, its role in heredity was not recognized until decades later. This discovery laid the foundation for modern genetics. 14. DNA replication is semi-conservative. Each new DNA molecule contains one original strand and one newly synthesized strand. This method was proven by the Mechelson-Stahl experiment in 1958. Semi-conservative replication ensures accuracy while allowing repair mechanisms to correct errors. It is a key feature of cellular reproduction. 15. DNA can be damaged by UV light. Ultraviolet light can cause thymine bases to form dimers. These distort the DNA helix and can block replication. Cells have repair mechanisms like nucleotide excision repair to fix UV damage. Excessive exposure without repair can lead to mutations and skin cancer. 16. DNA forms chromosomes. In eukaryotic cells, DNA is organized into chromosomes, which are long strands coiled around proteins called histones. Humans have 46 chromosomes, arranged in 23 pairs. Chromosomes ensure efficient packaging and segregation of DNA during cell division. They also regulate gene expression. 17. DNA is universal across life. All living organisms, from bacteria to humans, use DNA as their genetic material. This universality shows that all life shares a common biochemical foundation. It allows for techniques like genetic engineering and cloning across species. DNA's universality is key to understanding evolution. 18. DNA sequences can be compared to trace ancestry. By comparing DNA sequences, scientists can trace evolutionary relationships and genealogical history. Mitochondrial DNA is often used to track maternal lineage. Y chromosome DNA traces paternal ancestry. These studies reveal human migration patterns and evolutionary connections. 19. DNA can be modified in genetic engineering. Scientists can alter DNA sequences to add, remove, or change genes. Techniques like CRISPR-Cas9 allow precise editing of genomes. Genetic engineering has applications in medicine, agriculture, and biotechnology. It enables the creation of crops with desirable traits and gene therapies for diseases. 20. DNA contains both coding and non-coding regions. Coding regions, called exons, are sequences that code for proteins. Non-coding regions, called introns, do not code for proteins but have regulatory and structural roles. Non-coding DNA can influence gene expression, chromosome structure, and evolutionary processes. Both types are essential for proper cellular function. 21. DNA is tightly packed in the nucleus. Although DNA molecules are extremely long, they fit inside the cell nucleus due to tight packing. DNA wraps around proteins called histones to form nucleosomes. Nucleosomes coil into chromatin fibers, which further condense into chromosomes. This organization protects DNA and helps regulate gene expression. 22. DNA is complementary. The two strands of DNA are complementary, meaning A pairs with T and C pairs with G. This complementarity allows DNA to replicate accurately. If one strand is known, the sequence of the other can be deduced. Complementarity is fundamental for genetic fidelity and repair mechanisms. 23. DNA is involved in protein synthesis. DNA provides instructions to make proteins, which perform most cellular functions. Through transcription, DNA is converted into RNA, which then guides translation to form proteins. Proteins can be enzymes, structural molecules, or signaling molecules. DNA therefore controls all cellular activity indirectly via proteins. 24. DNA can form three-dimensional structures beyond the double helix. Under certain conditions, DNA can twist into supercoils or form loops. These structures help organize chromosomes and regulate gene expression. Some DNA regions can even form triple-stranded structures. These variations allow DNA to interact with proteins and adapt to cellular needs. 25. DNA replication uses specific enzymes. DNA polymerase adds nucleotides to the growing strand. Helicase unwinds the double helix. Ligase seals gaps between DNA fragments. These enzymes work together to ensure accurate and efficient replication of genetic material. 26. DNA can undergo repair. Cells have multiple mechanisms to repair DNA damage. These include base excision repair, nucleotide excision repair, and mismatch repair. Repair mechanisms prevent mutations from becoming permanent. DNA repair is essential for maintaining genomic stability and preventing diseases. 27. DNA can be methylated. Methylation is the addition of a methyl group to DNA bases, often cytosine. This chemical modification can regulate gene expression without changing the DNA sequence. Methylation plays a role in development, aging, and disease. It is a key aspect of epigenetics. 28. DNA can be sequenced. Sequencing determines the exact order of bases in a DNA molecule. Modern techniques like next-generation sequencing allow rapid and large-scale analysis. Sequencing helps identify genetic mutations, study evolution, and diagnose diseases. It is a cornerstone of modern genomics. 29. DNA sequences can be used in forensics. Forensic scientists analyze DNA to identify individuals. Even small amounts of DNA can provide a genetic fingerprint. This technique is used in crime scene investigations, paternity tests, and missing person identification. DNA evidence has revolutionized law enforcement and justice systems. 30. DNA is inherited from both parents. In humans, each parent contributes half of their DNA to offspring. Autosomal chromosomes are inherited from both mother and father. Mitochondrial DNA, however, is inherited only from the mother. This inheritance pattern determines family traits and genetic diversity. 31. DNA can evolve. Over generations, DNA sequences accumulate mutations. Natural selection acts on these variations, driving evolution. Evolutionary changes in DNA can result in adaptations to the environment. Studying DNA evolution helps scientists understand the history of life. 32. DNA can be artificially synthesized. Scientists can create custom DNA sequences in the lab. Synthetic DNA is used in research, medicine, and biotechnology. It allows for gene therapy, vaccine development, and creation of genetically modified organisms. Artificial DNA enables precise manipulation of genetic material. 33. DNA is negatively charged. The phosphate groups in DNA's backbone give it a negative charge. This property allows DNA to move in an electric field during techniques like gel electrophoresis. The negative charge also helps DNA interact with proteins and other molecules. Charge plays a critical role in DNA's biochemical behavior. 34. DNA can form hybrid molecules. DNA from different sources can be combined to form hybrid molecules. This process is used in recombinant DNA technology. It allows genes from one species to be inserted into another. Hybrid DNA is foundational for genetic engineering and biotechnology. 35. DNA can be damaged by chemicals. Chemicals like mutagens, pollutants, or carcinogens can alter DNA. Damage may include base changes, breaks, or crosslinks. Cells must repair this damage to prevent mutations. Understanding chemical effects on DNA is important for medicine and environmental safety. 36. DNA contains introns and exons. Exons code for proteins, while introns are non-coding regions. Introns are removed during RNA splicing. Exons are joined to form the final messenger RNA. Both introns and exons are essential for proper gene regulation and function. 37. DNA replication is highly accurate. DNA polymerases have proofreading abilities to correct mistakes during replication. This reduces error rates to about 1 mistake per billion nucleotides. High replication fidelity is essential to prevent mutations and maintain organismal health. 38. DNA is a blueprint for life. DNA contains all instructions needed to build and operate an organism. Every cell relies on DNA to function correctly. DNA determines inherited traits, development, and physiological processes. Without DNA, life as we know it would not exist. 39. DNA can form telomeres. Telomeres are repetitive DNA sequences at the ends of chromosomes. They protect chromosomes from degradation and prevent fusion with other chromosomes. Telomeres shorten with each cell division, which is linked to aging. Telomere maintenance is crucial for genome stability. 40. DNA can be used in personalized medicine. Genetic testing of DNA can guide treatment choices. For example, certain DNA mutations predict responses to drugs or risk of diseases. Personalized medicine allows targeted therapies for individuals. DNA analysis is transforming healthcare by enabling precision treatment. 41. DNA is packed into chromatin. DNA wraps around histone proteins to form nucleosomes, which further coil into chromatin fibers. This packaging compacts DNA to fit in the nucleus while still allowing access for transcription and replication. Chromatin structure is dynamic, changing during cell division or gene activation. Proper chromatin organization is crucial for gene regulation. 42. DNA strands are antiparallel. The two strands of DNA run in opposite directions, one 5-3 foot and the other 3-5. This orientation is necessary for replication and enzymatic activity. DNA polymerases can only add nucleotides in one direction, so replication occurs differently on each strand. Antiparallel structure is fundamental to the mechanics of DNA processes. 43. DNA contains genetic instructions for proteins. Genes in DNA encode sequences of amino acids that form proteins. Proteins carry out structural, enzymatic, and signaling functions in cells. The genetic code ensures that the correct amino acids are assembled in the right order. Errors in DNA can lead to faulty proteins and diseases. 44. DNA can form loops and supercoils. DNA looping brings distant regulatory elements close to genes, influencing transcription. Supercoiling compacts DNA and relieves tension during replication or transcription. Topoisomerases are enzymes that manage supercoiling. These structures help DNA function efficiently within limited space. 45. DNA can be replicated in a semi-conservative manner. During replication, each new DNA molecule has one original strand and one new strand. This method preserves the original genetic information while allowing replication. Semi-conservative replication was proven by the Messelson-Stahl experiment. It is fundamental to maintaining genetic fidelity across generations. 46. DNA is involved in epigenetic regulation. Chemical modifications like methylation or acetylation affect DNA accessibility. These changes do not alter the DNA sequence but influence gene expression. Epigenetic marks can be inherited or affected by environmental factors. They play a key role in development, disease, and adaptation. 47. DNA can be damaged by radiation. Ultraviolet, UV, light, X-rays, and other radiation can break DNA strands or form abnormal bonds between bases. Cells use repair systems like nucleotide excision repair to fix damage. Persistent damage can lead to mutations and cancer. DNA's sensitivity to radiation underscores its delicate yet resilient nature. 48. DNA sequences can be used for ancestry tracing. Mitochondrial DNA traces maternal lineage, while Y chromosome DNA traces paternal lineage. Comparing DNA sequences between populations reveals historical migration patterns. Genetic ancestry tests can identify relatives and ancestral origins. DNA provides a molecular record of human history. 49. DNA can be extracted and amplified. Techniques like PCR, polymerase chain reaction, allow scientists to make millions of copies of a DNA segment. Extraction isolates DNA from cells, while amplification makes it usable for analysis. PCR is essential for research, forensics, and medical diagnostics. It revolutionized molecular biology by making DNA analysis accessible. 50. DNA can mutate naturally. Mutations occur due to replication errors, environmental factors, or spontaneous chemical changes. They can be neutral, harmful, or beneficial. Mutations introduce genetic variation, which drives evolution. Studying DNA mutations helps understand disease and adaptation. 51. DNA is organized into genes and regulatory regions. Genes code for proteins or functional RNA molecules. Regulatory regions control when and where genes are expressed. Promoters, enhancers, and silencers are key regulatory elements. This organization allows precise control of cellular functions. 52. DNA can be methylated to silence genes. DNA methylation adds a chemical group to cytosine bases. Heavily methylated genes are often turned off. This mechanism helps regulate development and cell specialization. Abnormal methylation patterns can cause diseases like cancer. 53. DNA is transcribed into RNA. During transcription, RNA polymerase reads DNA and creates messenger RNA, mRNA. mRNA carries the code from the nucleus to ribosomes in the cytoplasm. This process is the first step in converting genetic information into functional proteins. It connects DNA to cellular activity. 54. DNA can be cloned. Cloning involves creating identical copies of a DNA segment or an entire organism. DNA cloning is used in research, medicine, and biotechnology. Scientists can insert cloned DNA into cells to produce proteins or study genes. It allows precise manipulation of genetic material. 55. DNA can be studied using electrophoresis. Gel electrophoresis separates DNA fragments by size using an electric field. Negatively charged DNA moves toward the positive electrode. This technique helps identify, compare, and analyze DNA samples. It is widely used in genetics, forensics, and molecular biology. 56. DNA is universal among living organisms. All known life uses DNA as the primary genetic material. This universality suggests a common origin of life. It also allows genetic engineering across species. Comparative DNA studies reveal evolutionary relationships. 57. DNA can recombine naturally. During sexual reproduction, DNA undergoes recombination, mixing parental genes. This increases genetic diversity in offspring. Recombination occurs during meiosis in eukaryotes. It is essential for evolution and adaptation. 58. DNA can be affected by chemical mutagens. Certain chemicals can change DNA bases, causing mutations. Examples include cigarette smoke, certain pesticides, and industrial chemicals. Cells may repair some damage, but persistent exposure can lead to disease. Studying chemical effects on DNA is important for public health. 59. DNA sequences can be edited with CRISPR. CRISPR-Cas9 allows precise modification of DNA sequences. Scientists can add, remove, or alter genes in living organisms. This technology has revolutionized genetics, medicine, and biotechnology. CRISPR enables targeted therapies for genetic disorders. 60. DNA is essential for life. Without DNA, organisms could not store genetic information or reproduce. DNA guides development, metabolism, and cellular processes. Its integrity is critical for survival and evolution. Understanding DNA helps explain the molecular basis of life. 61. DNA contains repetitive sequences. Repetitive DNA consists of repeated base sequences scattered throughout the genome. These repeats can be short, called microsatellites, or long, called minisatellites. They often play roles in chromosome structure and gene regulation. Repetitive sequences can also be used in forensic DNA profiling. 62. DNA can be circular in some organisms. While most eukaryotic DNA is linear, bacterial DNA is usually circular. Circular DNA is also found in mitochondria and chloroplasts. This form helps protect DNA from degradation and simplifies replication. It illustrates the diversity of DNA organization in life forms. 63. DNA can undergo horizontal gene transfer. Some organisms, especially bacteria, can exchange DNA with other species. This process spreads antibiotic resistance and new traits quickly. Horizontal gene transfer accelerates evolution and adaptation. It shows that DNA is not always inherited vertically. 64. DNA can form secondary structures. In addition to the double helix, DNA can form hairpins, cruciforms, and G-quadruplexes. These structures influence replication, transcription, and gene regulation. G-quadruplexes, for example, are rich in guanine and play roles in telomere maintenance. Secondary structures add complexity to DNA function. 65. DNA can be damaged by reactive oxygen species. Reactive oxygen species, ROS, are byproducts of metabolism that can attack DNA bases. Damage includes base modifications and strand breaks. Cells have antioxidant systems and repair mechanisms to counteract ROS. Persistent damage can lead to aging and diseases like cancer. 66. DNA is used in forensic fingerprinting. DNA profiling identifies individuals based on unique genetic markers. Short tandem repeats, STRs, are commonly analyzed. This technique is used in criminal cases, paternity testing, and identifying disaster victims. DNA evidence is highly accurate and reliable. 67. DNA carries non-coding RNA genes. Not all DNA codes for proteins. Some sequences produce functional RNAs like tRNA and rRNA. These RNAs are essential for protein synthesis. Non-coding RNAs also regulate gene expression and chromatin structure. They demonstrate that DNA has diverse functional roles. 68. DNA replication begins at origins of replication. Replication starts at specific DNA sequences called origins. Helicase unwinds the double helix at these sites. Multiple origins allow rapid duplication of large genomes. Proper initiation ensures complete and accurate replication. 69. DNA strands are held together by hydrogen bonds. The complementary bases are connected by hydrogen bonds, 2 between A and T, and 3 between C and G. These bonds are strong enough to maintain the double helix yet weak enough to allow strand separation during replication and transcription. Hydrogen bonding is fundamental to DNA stability and function. 70. DNA is affected by telomerase. Telomerase is an enzyme that adds repetitive sequences to chromosome ends. It prevents telomere shortening during replication. In stem cells and cancer cells, telomerase activity allows continued division. Telomere maintenance is crucial for genomic stability and longevity. 71. DNA is involved in epigenetic memory. Chemical modifications like methylation can be inherited through cell divisions. Epigenetic memory allows cells to remember their type and function. This is important during development, tissue specialization, and disease. Epigenetics shows how DNA can be regulated without changing its sequence. 72. DNA is used in genetic fingerprinting for wildlife. Conservationists use DNA to identify individuals and monitor populations. DNA helps track endangered species and prevent illegal poaching. Genetic data informs breeding programs and habitat management. DNA applications extend beyond humans to ecological preservation. 73. DNA can form triple helices. Under certain conditions, DNA can adopt a triple helix structure. This occurs when a third strand binds in the major groove of the double helix. Triple helices can regulate gene expression and have potential therapeutic applications. They illustrate DNA's structural versatility. 74. DNA can hybridize with RNA. DNA strands can pair with complementary RNA sequences. This occurs during transcription and in laboratory experiments like northern blotting. Hybridization helps detect specific DNA or RNA sequences. It is a fundamental tool in molecular biology. 75. DNA can be analyzed using microarrays. Microarrays allow simultaneous measurement of thousands of DNA sequences. They detect gene expression, mutations, and genetic variations. DNA is immobilized on a chip and hybridized with labeled probes. This technology revolutionized genomics research and diagnostics. 76. DNA sequences can be conserved across species. Some genes and regulatory elements are highly conserved through evolution. Conserved sequences indicate essential functions, like cell division or metabolism. Comparing DNA across species helps understand evolutionary relationships. Conservation highlights the universality of genetic information. 77. DNA is dynamic inside the nucleus. Chromatin loops, compartments, and nuclear architecture influence DNA accessibility. DNA is not static, it moves to facilitate transcription, replication, and repair. Nuclear dynamics are essential for proper gene regulation. Advanced imaging reveals DNA's complex motion in living cells. 78. DNA can undergo recombination during meiosis. During meiosis, homologous chromosomes exchange DNA segments. This recombination increases genetic diversity in offspring. Crossovers are essential for proper chromosome segregation. Recombination is a key evolutionary mechanism. 79. DNA can be analyzed using next-generation sequencing, NGS. NGS allows rapid and massive sequencing of DNA fragments. This technology can sequence entire genomes in days. NGS has applications in medicine, research, and evolutionary studies. It transformed the scale and speed of genetic analysis. 80. DNA can be used to study ancient life. Ancient DNA, ADNA, is extracted from fossils, bones, or preserved specimens. ADNA reveals evolutionary relationships, migration patterns, and species genetic diversity. Techniques must carefully prevent contamination and degradation. Studying ADNA bridges the past with modern genetics. 81. DNA can be used in personalized medicine. By analyzing an individual's DNA, doctors can tailor treatments to their genetic profile. This approach predicts drug responses, potential side effects, and disease risks. Personalized medicine improves treatment efficacy and safety. DNA-guided therapy is increasingly used in cancer, rare diseases, and pharmacogenomics. 82. DNA can be damaged by ionizing radiation. X-rays, gamma rays, and cosmic rays can break DNA strands or alter bases. Cells use repair mechanisms like homologous recombination to fix this damage. Unrepaired breaks can lead to mutations, cancer, or cell death. Studying DNA damage from radiation informs cancer therapy and radiation safety. 83. DNA forms chromosomal territories in the nucleus. Each chromosome occupies a specific region called a territory. This spatial arrangement influences gene expression and DNA repair. Chromosome territories help prevent tangling during replication and transcription. Nuclear organization is critical for maintaining genome stability. 84. DNA can be used for genealogical research. DNA testing helps trace ancestry, identify relatives, and reconstruct family trees. Mitochondrial DNA and Y chromosome markers reveal maternal and paternal lineages. Combining DNA with historical records provides insights into heritage and migration. Genealogical DNA testing is now widely accessible to the public. 85. DNA can be extracted from ancient samples. Even thousands of years old, DNA can survive in bones, teeth, and preserved tissues. Advanced techniques minimize contamination and amplify degraded fragments. Ancient DNA studies reveal extinct species, human evolution, and population genetics. These discoveries deepen our understanding of life's history. 86. DNA can be chemically synthesized. Scientists can artificially create DNA sequences in the lab. Synthetic DNA is used in research, gene therapy, and synthetic biology. It allows precise design of genes and regulatory elements. Synthetic DNA enables innovations like designer organisms and novel medical therapies. 87. DNA can be visualized with fluorescent dyes. Dyes like DAPI or SYBR green bind to DNA and emit light under specific wavelengths. Fluorescence microscopy allows scientists to observe DNA in cells. This technique is used in cell biology, diagnostics, and research. Visualization helps study DNA organization, replication, and damage. 88. DNA contains both essential and non-essential genes. Some genes are vital for survival, while others confer advantageous traits or redundancy. Essential genes are highly conserved across species. Non-essential genes contribute to variation and adaptability. This balance supports both stability and evolution of genomes. 89. DNA can interact with proteins for regulation. Transcription factors, polymerases, and histones bind DNA to regulate gene expression. These interactions control when, where, and how genes are expressed. DNA-protein interactions are essential for cell differentiation and response to signals. Misregulation can lead to diseases like cancer. 90. DNA is negatively supercoiled in cells. Negative supercoiling helps compact DNA and facilitates strand separation for replication and transcription. Topoisomerases manage supercoiling by cutting and rejoining DNA strands. Proper supercoiling ensures efficient DNA metabolism. Supercoiling also affects gene expression and chromatin dynamics. 91. DNA can be used to study evolutionary biology. Comparing DNA sequences across species reveals evolutionary relationships. Conserved genes indicate common ancestry, while variations show adaptation. DNA analysis helps reconstruct phylogenetic trees and evolutionary timelines. It provides molecular evidence complementing fossil records. 92. DNA can be repaired by multiple pathways. Cells use base excision repair, nucleotide excision repair, mismatch repair, and double-strand break repair. Each pathway addresses specific types of damage. Efficient repair maintains genome integrity and prevents diseases. DNA repair mechanisms are crucial for longevity and cellular health. 93. DNA contains regulatory elements like promoters and enhancers. Promoters initiate transcription, while enhancers increase gene expression. These elements control spatial and temporal patterns of gene activity. Proper regulation is essential for development and homeostasis. Mutations in regulatory DNA can disrupt gene function and cause disease. 94. DNA can form loops to bring distant regions together. Chromatin looping allows enhancers and promoters to interact across long distances. Loops are stabilized by proteins like CTCF and cohesin. Looping regulates gene expression efficiently. It illustrates DNA's 3D complexity within the nucleus. 95. DNA can be methylated or acetylated. Chemical modifications like methylation or acetylation affect gene accessibility. Methylation usually silences genes, while acetylation often activates them. Epigenetic modifications control development, differentiation, and disease. They can be influenced by environment, lifestyle, or diet. 96. DNA can be used in synthetic biology. Synthetic biology uses DNA to engineer organisms with new traits. Scientists design synthetic genes, pathways, or entire genomes. Applications include biofuel production, medical therapies, and environmental solutions. DNA's programmability makes it a versatile tool for innovation. 97. DNA can be used in CRISPR gene editing. CRISPR-Cas9 targets specific DNA sequences for editing. Scientists can correct mutations, disable genes, or insert new sequences. This technology revolutionizes genetic research and medicine. CRISPR demonstrates precise control over DNA function. 98. DNA can form G-quadruplexes. G-quadruplexes are four-stranded structures rich in guanine. They are found in telomeres and regulatory regions. G-quadruplexes influence replication, transcription, and genome stability. Studying them opens avenues for cancer and therapeutic research. 99. DNA is essential for heredity. DNA passes genetic information from parents to offspring. It ensures continuity of traits and species survival. Mutations or errors in DNA can affect inheritance. Heredity relies on DNA's ability to accurately store and transmit information. 100. DNA is central to life and biotechnology. DNA is the blueprint for all living organisms. Understanding DNA enables medicine, genetics, forensics, and synthetic biology. It drives research in evolution, disease, and biotechnology. DNA remains the foundation of life and scientific innovation.